So we're going to move on now to Gang Fang from Mount Sinai, who's going to talk about DNA methylation. Take it away.
Thank you, Kia. Thank you for this opportunity because not just to share our research, but also to discuss this broad field of epigenomics and epigenetics with this education and outreach like audience that is broader. So this is who we are. So we develop technology to study epigenome, but also study epigenetic regulation. And we like very much enjoy studying human genome, but we also study some bacteria and microbiome. And as I will show you towards like through this talk, this actually gave us some unique perspective to better study the human genome. So thanks to lecturer who just already talked about a lot of backgrounds of epigenetics of human, and especially with the chromatin and histone modifications, I will directly like focus on the DNA methylation. And as you'll see in this talk, I will still talk a lot about the epigenomic technology as in the first session. So if you enjoy some of the cutting edge technologies, and you'll still get some flavor of that in my talk. So when we talk about DNA methylation, actually 99% of the cases, we are specifically referred to one form of DNA methylation in which the methyl group is added to the 5th position of the cytosine. And in short, we call it 5MC. And this is very important, not only because it's very abundant in human genome, but also it's play very important roles in the development and also in human diseases like cancer and brain. However, if we step back a little bit and see this 5MC in the whole universe of DNA chemical modifications, this is only one piece of the whole universe, because even just on cytosine, you can have other kind of methylation, like not just 5MC, you can have hydroxymethylcytosine, or even further additional derivation during the demethylation process. And the important thing is, although if you just look at these like letters, it looks very similar to each other, but actually their biological functions are very much different, and it's very important to differentiate between them. And a methyl group can also occur on the 4th position of cytosine, in which we call 4MC, and it can even occur on the adenine base, which is like completely different from cytosine. For example, we have like 6MA. So now with all of these, we have basically I use green and red, 2 different colors to mark. Like some of them are very much unique to humans. Some of them are very much unique to bacterial. But one of them, which is 5MC, is really shared between human and bacterial. So in addition to this methylation, we also have all kinds of DNA damages that can respond to environmental perturbations and some of the stresses, and some of them can cause mutations. So the goal is then to how can we effectively map these modifications on the human genome. And some of you may heard about this very simple technology called bisulfite sequencing, but for those who may not have been heard of this, very simple explanation is that if you have these this original DNA as shown here, and these 2 Cs have this methyl group, but this one and this one doesn't have a methyl group. If you add this bisulfite, it will convert those non-methylated C into U base, but it will leave intact the original methylated Cs. So now if you sequence these converted DNA, then basically you can tell which one hasn't been converted. It indicates there's some methylation out there. So this is very much widely used, but it creates DNA fragmentation, and also it does not really differentiate between 5MC and 5HMC very well. So this motivated a lot of work that can better differentiate between them. And this is just one of the recent examples called TAPS sequencing. And you don't need to like worry about all of these chemical compound names. Basically, through the use of combinations of these chemicals, you can actually differentiate between different DNA modifications. And remember that in the whole universe of DNA modifications, there's like different kind of DNA modifications as shown now as the columns. And accordingly, like scientists have becomes very creative and develop different kind of approaches, chemical approaches, or enzyme-based approaches, or antibody-based approaches to map like different ones of these modifications. And the process that they're in general very specific and sensitive, and many of them can also provide you base resolution analysis, and importantly, many of them are quantitative. So these are really great, and these are the cornerstones of a lot of our understandings about epigenetic regulation in the human genome. But at the same time, these kind of approaches also have some limitations, which is if you want to make great use of these protocols, even after the paper got published, you still need some experience. You need to like optimize it. And because the field is like evolving very fast, so when a new and better protocol got developed, almost always you need to optimize again. And if you want to study different kind of modifications, then you need to do separate assays to really probe them. And because of all of these limitations, in most studies, people focus on only one or maybe like a couple of modifications, but not really comprehensively. This motivated the field to go towards like newer generations of sequencing, basically the third generation sequencing, represented by nanopore sequencing and pachybiome sequencing. So, and one of them, I will show a quick example. This is called like one of the technology called smart sequencing. In this video, you can see a DNA polymerase is doing real-time DNA synthesis, and it's reading the template strand. And if you see a color, that means there's a new base incorporation that match, like complement the template strand. So, and different colors reflect different bases. But the most exciting thing is not just these colors, but these gaps between different colors, because this, what we call the time dimension, can tell you how long does the DNA polymerase take to translocate from one base to the next base. And why this is important is because, like in this example, these are two exactly the same DNA sequences, but the only difference is that this editing is methylated, but this editing is not methylated. So you can see it takes much longer for the DNA polymerase to translocate from this editing base to the next one if it is methylated compared to this unmodified editing. And imagine you can calculate simple ratio that you can basically see very pronounced signal-to-noise ratio right on top of this modified editing. And so the other one is nanopore sequencing. I will not get into the details because in the first session, Mark and Sarah and even Kaya in this introduction have already introduced. Basically, nanopore sequencing is really fantastic. It's provided another way to read DNA modifications directly from the sequencing of native DNA. So I will only provide like this very high level view. So about 10 years ago, we used smart sequencing to first map to study 6-man 4MC in bacterial. And so at that time, smart sequencing is not really great for 5MC. And really interesting is the other case, that nanopore sequencing is doing really great for 5MC mapping in human, but initially is not doing as great job for 6-man 4MC detection. But luckily, scientists, again, are very creative. And nowadays, basically at the high level summary, like both of these technologies can do like fairly well for both of these three, for all of these three kind of modifications. There's still some like subtle details that like some have strengths, some have weakness, but at the high level, which people have different choices depending on different projects, people can really have choices to tailor. And so these are the uniqueness of third generation sequencing that allow you to directly map these modifications without all of those chemical approaches. But remember, these third generation sequencing also have the unique ability to generate much longer reads compared to the short read sequencing. So what that means is almost like a puzzle. If you want to piece together some very complex puzzle, then of course you want to play with bigger pieces. And this leads to a great motivation why this matters for the study of the human genome is because nearly 50% of the human genome actually have one way or another kind of repetitive elements. So it's very important to use long reads to better resolve them. And some of you may have already heard the exciting news about the T2T consortium recently. There's a special issue in Science. And basically this cover image shows that all of these red colors are those extremely hard regions that cannot be resolved using traditional short read sequencing, but they can be resolved much better using long read sequencing. And you can see that the human genome really become complete. But what we feel even more excited is that not only the genome becomes more complete, but because of these long reads, you can actually study the demethylation in those regions. And that's exactly why 3 papers in this issue by like 3 different groups actually for the first time can really ask and answer questions that what's really going on in terms of DNA methylation status in these kind of very complex regions. And to give you some even more idea about a specific example, this is one of the case that is one kind of repeat called short tandem repeat. So this is a gene that is linked to a disease that is well known called ALS. And inside this gene, there's like G4C2, basically 4G and 2C, and basically a lot of copies out there. It's very hard for short reads to resolve it. The long reads can resolve the sequence and also methylation, which is makes very major difference. And this is another case. The KRT19 is another gene that is playing an important role and upregulated in breast cancer. So not only long reads can tell you in complex sequences, it can even tell you which allele of the two chromosomes have methylation. And in this case, it is this allele 1 have this methylated CPG in here, but actually not in the allele 2. Basically, you can also do the phasing of demethylation onto the long reads. So now I remember this slide, we're talking about the whole universe. There's so many different kind of modifications. So if third generation sequencing can give people opportunities to find directly the modifications, then not surprising people want to apply this technology to go beyond 5MC in the human genome. And the 6-MA is one of the main targets because it plays a very important role in bacteria. So naturally, people want to ask whether or not 6-MA also exists in human genome and whether it plays very important roles. And why is another reason is if you just think about 5MC, one form of modification, people find so many great roles and it play in the human genome. So if people really can discover one additional form of the modification in the human genome, that really provide a lot of new dimension to study both like human health and human diseases. So encouragingly, I mean, initially about 7 years ago, some papers published that 6-MA exist in eukaryotes, specifically in unicellular eukaryotes. And these are the examples that previously people know they exist, but they cannot map them. So this one is really third generation sequencing can allow people to map them. So, and then quickly, almost like 1 year later, so people study all kinds of multicellular eukaryotes. So you can name all the model organisms: C. elegans, Drosophila, plants, and vertebrates, mouse, and human, of course. And within human, people study different kind of disease and different developmental stages, different tissues. So initially, a lot of excitement really kind of emerging because, as I said, one additional base modification in the human genome really opens a lot of new opportunities. So, however, some like unexpected things happen, which is some papers started to raise some concerns and doubts that there's different kind of confounding factors and there's different kind of contaminations and there are different kind of lack of sensitivity. What if there's some like RNA contamination, or what if there's some false positives? So people raise these kind of doubts and there basically leads to some debates. However, as you see, even after these initial papers that raise these issues, still some papers get published and reporting even more exciting discoveries, even more specific mechanisms behind the 6-MA in the human genome or the mouse genome. So you may feel a little confused why like people for the same question can have so much like contradicting answers. This is actually the reason, which is the key challenge is that actually no method can reliably and quantitatively and specifically map 6-MA on eukaryotic genomes. And it sounds a little strange, but actually that's the reality because like sometimes these hypotheses occur before the technology is ready. So that's why we really need to be cautious in the use of these cutting edge technologies on real problems. So in this case, I will provide a very simple like story, which is what we did is we use that third generation sequencing called smart sequencing. The one uniqueness of this sequencing is that you can read one molecule, the two strand, again and again, like even like hundreds of times. The reason is because you add this adapter that is like a smart bell, and this polymerase read basically can read again and again during this. This gives us a lot of confidence on what's really going on this molecule, that we can have much higher signal to noise ratio. But the second thing that we did was like we do not want to only look at the human genome, because if some people raised concern that there may be some bacterial contamination in the genomic sample and bacteria have very abundant 6-MA in their genome, then we really want to examine everything. So what happened was that we sequence everything and we analyze everything, not just the species of interest, but also different sources of contamination. And then we train a machine learning model that can tell us what's the abundance of 6-MA on each of these different components, so that we can really tell where does this 6-MA come from in a genomic sample of interest. So initially, what we did was we studied the unicellular eukaryotes, and remember the 6-MA in them are real, and people just didn't know where they are. And we actually find very interesting discoveries that even between two like unicellular eukaryotes, the 6-MA distribution are actually very different. And in one of them, which is basically, you don't need to worry about this complex name, basically it's called green algae, 6-MA is very much located specifically towards the nucleosome boundary. So there's even some hypothesis about maybe 6-MA can help the nucleosome positioning in these early eukaryotes. And in some sense, this also linked to Lacroix's like first talk that whether DNA modifications can like have some crosstalk with the chromatin interactions and the chromatin states out there, right? The next one, what we applied to is in Drosophila. So Drosophila, what we did was there's a paper like 7 years ago. The discovery reported there was that 6-MA is specifically high in early embryo, like even before the first hour, like maybe just after 45 minutes. So what we did is we collect the early embryos after 45 minutes from Drosophila, and we use this approach to sequence it and want to analyze it. So the first observation that we found is, yes, almost everything are Drosophila, but about 3% are actually come from microbes, some from bacterial, some come from yeast. And so using our approach, remember that we can individually and separately estimate the abundance of 6-MA on these different sources of DNA. So the first thing you see, in total, we indeed observe a lot of 6-MA, like 100 ppm. ppm means 1 out of a million. So 100 ppm means 1 out of 10,000. That sounds small, but actually for new modification, that is pretty abundant. But if you look at individual abundance in Drosophila, you can see it's much, much lower compared to the total, because the vast majority of 6-MA actually come from these two bacteria. And if you consider these density of 6-MA and also the genome size and the abundance of the different species, actually the 6-MA in the Drosophila only explains about 1% of the total 6-MA in Drosophila early embryo. So this is a very kind of helpful discovery for the field. And the next, we applied to some other.
5 minutes left.
Okay, thank you. So the next what we applied it to is to the human and some other eukaryotes, but I will highlight the human here. Initially, we examined the positive controls, and clearly these as expected very high, like super high. But then when we applied to a PBMC, like blood cell samples, and also to glioma tissues, which were reported to be very high, like in the previous studies. And actually, we see it's much lower than previously reported. And different from the case of Drosophila, we didn't see contamination in our samples, but the levels are still much lower. So we were basically thinking, what could be the reason? We do not have access to original samples, but we have some clues. One of them is that because sometimes people do genetics, they use plasmids, and the plasmids actually were replicated in E. coli that actually have a very abundant 6-MA. So basically, those plasmids that you use in all of these genetic experiments will carry a lot of 6-MA on them. So that could be a source of external 6-MA. And we did an experiment that's exactly as we expected. So on human, it's actually, again, very low, but most of the 6-MA come from this plasmid called PCI, and also there's some residual amount of E. coli, even after you clean up. Basically, some kind of residual amount of bacteria is very hard to avoid. And similarly, we estimate the abundance of 6-MA in human that in this example, again, is a very, very low abundance. So This may sound a little bit discouraging, but actually it's not, because when we work on new and cutting edge problems, actually this is very common that there's some kind of confounding factors is actually very common. But another reason that people should not get discouraged is because the next slide, which is even if a modification is not present in a genome, say in human genome, people can still be very creative to use it as an exogenous way to mark things on the human genome. Like this is a very simple example. Like this is blue circles represent nucleosomes, and these are like open chromatin. So imagine that if this DNA is wrapped around this nucleosome, methyltransferase cannot add 6-mA to the DNA. But if it is open chromatin, you can add the 6-mA onto this open chromatin region, and then if you read it out, and remember this long read sequencing can read long pieces of DNA, you can actually phase this open chromatin status on individual molecules across 10 kb. So that's super exciting. And quickly, scientists becomes very interested in this. There are already 4 papers that all did fantastic job. And in this figure at the bottom, basically each line is what they call a fiber. Basically, it's an individual molecule, and from the color you can tell which region is open chromatin. So super exciting. And the most recent work just published 2 months ago by a group probably in Lakehurst department. I mean, they can even use this to study protein DNA interaction. So I mean, the final summary that I have is actually very encouraging, because still demethylation plays a very important role in human genomes, so it's very much worth studying them. And third generation sequencing provides great opportunities, both in terms of the direct mapping of demethylation without chemical approaches, also in terms of that it allows people to study demethylation in repetitive regions. And finally, we want to highlight both the prospects and the pitfalls in mapping novel forms of demethylations. And it's very common to have some challenges, but I definitely believe in the future, like these technologies will help people study the human genome in a much better capacity. So I'd like to thank my group on the left here in our group, especially Imeng Kong, and also our colleagues in the genomics department, Ginter's and Robert, and our collaborators at Rutgers University, University of Chicago, and Emory, and like Chinese Academy of Science, and of course to the funding agencies. Thank you.